Hello, my dear pharmaceutical colleagues. Pharmaxis 0007 under the banner of Manoj Creation 0007 brings to you knowledge enrichment sessions and programs. Today's discussion and elaboration point will be on the media filling simulation study basic data, its purposes and acceptance criteria. The Pharmaxis 0007 is basically involved in the demonstration and exhibition of the pharmaceutical and healthcare snippets, which will cover the basic database of any concept in the pharma and healthcare domain. This concept will be in a very consolidated and simplification manner, which will help all of us as a professional and as a newcomer in the industry for facing the audits and compliances, as well as the interviews. There will be detail on the guideline exposure in the subsequent forthcoming sessions, which will be covered under the banner of Pharmaxis 0007. Keep watching, subscribing, and like will be welcome from your end. This will be a concept-based series which will be covered in the forthcoming next 10 sessions, and will be covering a latest in industry concept and demonstrations. Any question and answer can be elaborated or defined and posted back for me to answer you back. Okay, so keep watching, sharing, and subscribing. As a database for the initial, we should know some basics of the media fill simulations. What are the batch sizes and what is the actual batch size that need to be simulated for the aseptic simulation study. What are the acceptance criteria? What is the action that need to be taken in case of any failure mode as action level number 1 and subsequently action level number 2. As a thumb rule, there should not be any contaminated unit in the media filling study, which will demonstrate your process being a very robust, that is repeatable and reproducible without any contamination across the process execution pattern. Now, when we talk about the batch sizes, the batch size should be either less than 5,000, between 5,000 to 10,000, or greater than 10,000. These are the commercial batch sizes which have been executed across the pharmaceutical industry. But when we have to design for the simulation study, we cannot go for the actual batch sizes, but we have to have some justified rationale which will define the batch sizes. So we have mentioned in the slide also, for the less than 5,000 batch size, the batch size should be at least or equal to the production batch size. For the batch size of 5,000 to 10,000, the batch size should be of comparable size to the production batch size. And for the batch size of greater than 10,000, the batch size can be the most accurate simulation model would be full batch size and the duration that need to be covered. Now, as we said, that there should not be any contamination in it. But sometimes due to external and internal environment and the process controls, there might be some failures which need to be addressed after its observation during the study, or after the results are, or after the contamination pattern has been identified. So for the batch size of less than 5,000, if there is a failure of 1, then action level number 1 indicates it is a cause for revalidation. For the batch size of 5,000 to 10,000, if there is a contamination of 1 unit in this batch size, then the action level number 1 states it is an investigation of that failure mode, and action number 2 states that it is a consideration for repeat media fill. Now, for the same batch size of 5,000 to 10,000, if there are 2 failures, 2 contaminated vials or contaminated ampoules, then the action level 1 states it is a cause for revalidation, and action level 2 states that it is an investigation. Now, for the batch size of greater than 10,000, the most accurate simulation model which we have said should be a full batch size, which is practically not possible, but the duration has to be covered and appropriateness of the batch size that has been designed for this study of the batch size greater than 10,000. Now, if there is a failure of 1 contamination or contaminated unit, then the action level 1 state it is an investigation is required to be done of the failure mode. And if it is a batch size of greater than 10,000 and with a failure of 2 contaminated units, then a revalidation is action level number 1, followed by cause for revalidation following investigation is action level number 2. Now this is as regard the database or the systematic approach towards the understanding of contamination and no contamination. Now let us try to understand in depth the purpose of this particular media field simulation. The first parameter that is to be considered is the capability and reproducibility. Why media filling is required? Media filling is required across the process line as a validation parameter for understanding the capability and reproducibility, that is to assess the capability of the aseptic process to produce sterile product reproducibly. The second factor is contamination risk assessment. Here we assess the contamination risk factor of the process and hence the state of control is identified and justified. The third is microbiological assessment. This assesses vulnerability to microbial contamination. Fourth is the practices and procedures demonstration. Here we need to demonstrate that the aseptic operating practices and procedures are appropriate in line with the process design. Next is personal practices. The human factor plays a vital role in the evaluation of the process line. Hence here this covers the evaluation of the aseptic processes, personal practices, whether to qualify the personnel, whether to requalify the personnel, or whether to disqualify the personnel. And the last being the CGMP approach, then compliance with good manufacturing practices and regulatory expectations. Now all this database and detailing done across this slide has been covered under the FDA guidance for industry, that is sterile drug products produced by aseptic processing, current good manufacturing practices of September 2004, PDA technical report 22, process simulation of our aseptically filled products 2011, EU GMP Annex 1, which is lately revised on 25th August 2023, it is manufacturing of sterile medicinal products, and USP chapter 797 covers all the detailing on the media film aseptic techniques. So my dear colleagues, please share, subscribe or feel free to comment for any provisions or revisions or latest update under this global regulatory pharma guideline for us to enrich each other. Basically this banner indicates what is right, what is wrong, what is the choice, what is the decision made under the 5 headings of the GMP aspects, that is man, machine, material, method, market. Whether we are right in the process, we are wrong in the process, we are what choice to be implemented to proceed further, and what decisions to be made. So this is all on the media filling basic data of purpose and the acceptance criteria from Pharmexus 0007 under the banner Manoj Krishnan 0007. Do visit the website, do visit the web page. Thank you all for your concentration. Thank you.